Okay, so definition of a gene. I better have got this one right. Okay, so we've got sequence of... I didn't know whether it was bases or nucleotides. It doesn't matter. I said nucleotide bases. In DNA... Oh, did I not mention DNA? It was so obvious to me that codes for the primary structure of a protein allow protein. Well, let's rewrite that. I think I'm going to take myself off this. See, I didn't mention DNA, but I mean, it was so... Oh. That's why it's so important to write these definitions out. And I'm going to write it out as 2 bullet points. Well, okay. No, I'm not, because I don't have space. Codes for primary structure. Oh, can't believe it. Can't believe it. So I got the mark for the protein. Okay, what have we got here? The role of tRNA in the production of leptin. Okay, I'll read my answer. There's basically nowhere I can put this. But you're basically fully zoomed out at this point. The tRNA anticodon is complementary to the messenger RNA codon. They form hydrogen bonds with A with U and C with G. Ribosome holds 2 transfer RNAs in place at a time. Each transfer RNA carries an amino acid that is specific to its anticodon. Peptide bonds form between the 2 adjacent amino acids, adding an amino acid to the polypeptide chain. Transfer RNA returns to the cytoplasm to collect another amino acid. Okay, so we have tRNA molecules transport amino acids to the ribosome. Carries. I don't think I actually said that. Transports. I'm going to do alpha. I don't think I actually... Each one has an anticodon. Binds to the codon on the messenger RNA. Has an anticodon complementary to the mRNA codon. Each tRNA carries a particular amino acid. Each tRNA carries an amino acid that is specific to the anticodon. Again, another one that we need to just add to my mental notes. I mean, it's so frustrating. That's why verbal exams for me would be way better, because they would actually assess someone's ability to do the subject. Okay, so what have we got here for this hydrophobic stuff? Primary structure sequence determines the folding of the protein. Position of the R groups determines the tertiary structure. Forming a globular structure. I said arrange themselves. I mean, if you want me to mention tertiary structure, describe how the primary structure and tertiary structure, because it definitely was misleading, that question. The primary structure of the amino acid determines the folding, position of the R groups. The primary structure of the leptin protein is the sequence of amino acids. Each amino acid has a different R group, which has different charges. Determines the sequence. Primary structure of the sequence of amino acids determines the folding, is the bit that the allow the position of R groups. The sequence of amino acids, each was the sequence of amino acids. each amino acid has a different R group. So I mean, between the two sentences, it's probably okay. Didn't say forming a globular protein. Hydrophobic R group is located in the center, hydrophilic on the outside. Hydrophilic R groups on the outside. Water forms hydrogen bonds with the hydrophilic groups. Dipole, hydrophilic interactions between the water and the protein. The sequence of amino acids determines the folding of the polypeptide. It doesn't say what, the position of the R groups. I think the position of the R groups is what it means for this part, which is the mandatory part. Each of which have different charges. I think that's just sneaking in for marking point 1. Which gives 2, but I'm going to read this is a good answer to rewrite. Again, if you can just do it closed book, so you can like cover this over. Okay, so that page was -1. What do you think? Should I give myself that mark? It's unclear as to whether position of R groups, and it must be the bit that's outside the brackets. I would assume. Okay, so frame shift question. Let's just look at the mark scheme first of all. Adding or removing a 2 changes the triplet code. Different codons are produced. Adding a new start or stop codon. Coding for a shorter sequence of amino acids. Changes the okay, let's bring it in full zoomed out. Changes the DNA triplets. Changes the DNA triplets code is what it wants. Allow different, if a codon is changed, which is also different codons produced, coding for amino acids into a change from an amino acid into a stop codon, coding for a shorter sequence. Translation will be stopped prematurely. Translation stopped. Shorter protein. Okay. So I dropped 1 on that page. I dropped 2 on that page, so 3 total. 7 out of 10. 3 marks down.